intro. I don't have too many other things to add. Yes, so the project I'll talk about briefly today is utilizing data from the ARCH cohort, so really the pilot to the larger cohort currently being recruited. But my overall focus is on maternal nutrition, also environmental exposures. And today I'll talk about other work we do related to maternal obesity. A little bit of what I'll talk about today is a little bit more mechanistic. Our group tends to be really interested in sort of how and why things happen. So we, try to understand not just the implications of, women who are obese for their offspring and for their own health, but also what could be potential mechanisms of this. And so we tend to collect a lot of biospecimens. We work with cohorts and groups like this one that do also collect a lot of kind of biospecimens. And so it's really important for us to be able to connect that mechanistic side of things to the health outcomes. So the project today will focus on mitochondria, and I have these cute little cartoons on the front just to say that mitochondria is really important. I'll talk a little bit about their function, but essentially we got them from our mamas. And so this was, I think at some point I used this slide right around Mother's Day, so it was kind of perfect. We're a little early this time around. So let me make sure I can do this. So this project includes 4 of us. 2 of us are here at Michigan State and 2 are at the University of Michigan. So it's a cross-campus collaboration. 2 of us are here doing some aspect of the project, and then the folks at University of Michigan will continue on with the project once we're kind of done with our parts. So our work for this particular project is focusing on maternal obesity. And just to give a really brief introduction, often we are concerned if women are gaining too much weight in pregnancy or not enough weight in pregnancy. But one other important factor that we consider often is whether or not they come into pregnancy being overweight or obese, because we know that that puts them at greater risk. So in this case, we're sort of thinking about overweight being the 25 to 29.9 body mass index here in yellow, and then sort of anything past that we consider obese in various categories of obese. So this figure essentially shows the estimated prevalence of maternal obesity in the United States. These figures are a little bit older. They came from a really nice review. The numbers have not decreased, unfortunately. So the figures are very similar now. And essentially what it shows is that kind of the darkest purple line, I believe, that is showing us the overall prevalence across all racial sociodemographic categories. And it falls, this is almost 10 years ago, was it around 30? I think we're probably closer to 35, 36, women being obese specifically. Overweight and obese is more like 50, 55% currently. One other thing to notice is that the prevalence they're not equal across all racial categories. So for example, non-Hispanic Black women actually have much higher prevalence of obesity when they're actually going into a pregnancy. And one important thing for us is that here I've circled the 30 mark, the 30% mark, and this is about the prevalence of obesity in the ARCH subcohort, so the group that we're working with specifically. So it's a really nice population in terms of it being very generalizable to the overall population. So when we're trying to ask questions related to, what are the implications of having women who are going into a pregnancy obese, or what are the mechanisms, we can hope that some of those answers would be generalizable to the rest of the population. So why worry about obesity in pregnancy? And rather than kind of my usual list of things, I've used this nice graphic. Again, 2 in 3 women are either going into a pregnancy either overweight or obese. We know that maternal obesity increases the risk of various childhood metabolic dysfunctions, including obesity, metabolic disease. And then this sort of functions through various mechanisms. A lot of it has to do with placental nutrient delivery. A lot of my graduate work was actually focused on placental metabolism in women who are overweight or obese compared to normal women. And that process is very different because obviously the amounts of nutrients and the types of nutrients being transferred to the fetus are quite different when there's an oversupply of glucose. There's also quite a bit of inflammation that happens in women who are obese during pregnancy. Pregnancy happens, or portions of pregnancy are classified or characterized by increased inflammation already. And so having that additional adipose tissue and having that additional inflammation is also part of the issue. The epigenetics piece is really important. That's sort of how we think that the message is essentially being transferred to the child through epigenetic mechanisms. And then maybe you've heard a couple of talks, Dr. Comstock, Sarah studies the microbiome, and so do other researchers at MSU and also at U of M and at Wayne State. So they're interested in how various factors, including maternal obesity, can actually impact the maternal microbiome, and then also in that sense, also impact the child's microbiome. So again, our group is sort of interested in that kind of mechanism realm. And one of the things that I suddenly wanted to ask about, and this cohort was perfect for that, is this idea of maternal mitochondria. So back to sort of high school biology days, this is a cell, and the mitochondria are those little squiggly things, round with squigglies on the inside. I've kind of circled a couple or one of them in red, and you can see there are actually multiple mitochondria within this cell. So what's so important about mitochondria? We know, again, back to high school days, the definition is that the mitochondria is the energy source of the cell. It's really critical for all aspects of cellular health. And this kind of describes all of the other functions of mitochondria. And what's really important is that they can be functional or they can also be dysfunctional. And so what we were interested in is trying to understand whether or not women who may come into pregnancy overweight or obese are kind of leaning towards the dysfunctional range. So potentially whether or not they have higher inflammation, whether they have cellular death. And so all of these things that are related to maternal health. I've also have this slide here that sort of talks about how this may be happening, that here at the top are all the defining characteristics of obesity. We tend to have higher free fatty acids in circulation. So for example, if a woman who is pregnant goes those very early maternal visits, the perinatal visits to the OB, the triglyceride levels tend to be very, very high. Cholesterol tends to be elevated. So hyperglycemia, also glucose levels tend to be elevated. And all of that has some impact on mitochondrial function and dysfunction. And that in return can potentially lead to sort of the same dysfunction. So it's almost like a cycle. And so ultimately there's potential for additional insulin resistance, cardiovascular disease, and various other issues. So one way that we can think about, okay, so does somebody have a healthy mitochondria? Do they not have healthy mitochondria? Is we can actually count them. We can use certain methods, and I'll show really briefly, to essentially count what kind of or how many mitochondria somebody has. So for example, here I'm showing, again, this is kind of pointing to that squiggly organelle in the cell. And it's interesting, we always think that our DNA is found in the nucleus, but actually mitochondria have their own DNA. Each of those little mitochondria have their own. And so when we count mitochondria, we can actually tell how many when we count their DNA or mitochondrial DNA, we can actually essentially predict how many mitochondria somebody has. And we kind of assume, this has been suggested that sort of the more mitochondria or the fewer mitochondria you have, it's a good biomarker of your metabolic health. It's sort of an interesting biomarker of that. The folks at University of Michigan are also interested in the epigenome. So one of the things I mentioned was that we think that one of the things that sort of predicts and programs child health and fetal health is the epigenome. Well, we know that since mitochondria have their own genome, there's a possibility that it could also be impacted by their epigenome, which is kind of the regulator of that process. And so that's something that they're interested in. So again, yes, also in the mitochondria. So the main goal of this project is just to really sort of not simply evaluate whether mitochondrial endpoints differ in obese versus normal weight pregnant women. And specifically, we'll ask 2 questions about mitochondria. We'll count them. So we'll ask how many mitochondria do overweight and obese women have compared to normal weight women. And then we'll also do some epigenetics at the University of Michigan and specifically focus on DNA methylation, which we know is really important. So I know that this isn't the process that the current cohort is taking, but one of the really beautiful things about the ARCH cohort was that they collected maternal blood spots. So not just the blood spots that were collected as part of the state's sort of baby, or not just the blood spots collected from the baby from the state, which is also sort of beautiful. I had my first here about 6 months ago, and I gave consent for someone to use my blood spots for research. It's really lovely. I come from a state that doesn't do that. So it's sort of amazing. So the study actually collected maternal blood spots. And so we decided to try to figure out how to use those because they were sitting around not being used. And so we decided to try to actually collect DNA or extract DNA from those blood spot samples from moms. So we are essentially isolating DNA from those maternal blood spots. We've just now tested whether there were some that were stored in the freezer, there were some that were stored as in the room temperature. Both were relatively consistent. We decided to stick with the frozen ones, but it's sort of incredible that even spots of blood that were sitting at room temperature have relatively great function structure, all of the good DNA structure. We're going to use PCR to essentially identify how many mitochondria someone has, and then we're going to use another technique to look at DNA methylation. So why is this important? We know that it seems like these sort of mechanistic questions are like just digging deep, but we understand that hopefully if we know more about how maternal obesity impacts the mother, the fetus, we can potentially create targeted interventions. We can try to understand which women are more or less susceptible. And that is part of the future direction. So one of the things that we're really interested in is to try to understand whether there are women who are more susceptible, whether we can use something like mitochondrial copy number as a biomarker of, again, looking at susceptibility, maybe sociodemographic, racial, maybe maternal age, which we know is an important predictor of copy number as well. So those are some of our future directions. And that's everything that I have today. And I think